All right, we're going to nerd out on some microbiology with artificial intelligence and machine learning. There's really two major areas where machine learning and artificial intelligence is really taking off. One of them, financial services, a lot of things through anti-fraud. The other one, healthcare, life sciences, pharmaceuticals, drug discovery, doing some incredibly exciting things. And I haven't found anything more interesting than what this study has done. Molecular de-extinction of antibiotics from ancient proteomes using deep learning. And look at this graph. De-extinction. What the heck is it talking about? Some Jurassic Park type crap? What it's doing is using historical databases of proteomes and analyzing their sequencing in order to see if there's anything that will help us develop antibiotics for today. Because generally, what we deal with is forward-looking. Antibiotic resistance is a major problem. As a biochemistry undergrad, I studied virology and microbiology, and we talked about this. The overutilization of things like hand sanitizers. I mean, through the pandemic, this was something that I was personally worried about, and what a lot of people worry about, in both virology and microbiology. We continue to create antibiotics that attack these things, and what's left over are the more resilient and evolved microbes or viruses that we have to continually create additional vaccines or antibiotics to attack. And as we continue down this path, you're going to get the ones that are the strongest that survive. So what this study aims to do is, again, instead of a forward-looking approach, let's look at the backward approach. Is there anything within the proteomes of these extinct animals that we can rely upon in order to come up with other antibiotics? So in looking at the abstract, broad overview, I don't want to go too in depth, just kind of tackle the more interesting pieces of this. The abstract first sentence: molecular de-extinction aims at resurrecting molecules to solve antibiotic resistance and other present-day biological and biomedical problems. And this is what they found. So from the proteomes, and actually there's these things that are called encrypted peptides. And I believe I have, here we go, a University of Pennsylvania article, newly discovered encrypted peptides with antibiotic properties. So these are things that are studied within humans. There's some peptide sequencing that we can rely upon to create antibiotics. This is what they're looking for through the extinct database of the DNA sequencing. So we go back, and this is what they found. Most peptides, the encrypted peptides that they found in the extinct animal sequencing, killed bacteria by depolarizing their cytoplasmic membrane, contrary to known antimicrobial peptides, which tend to target the outer membrane. Targeting it, when you look at something like how antibiotics attack a microbe within us when you swallow a pill, it's going to look something like this generally. It's going to attack the outer wall, the cytoplasmic membrane of a microbe, make it disintegrate, disappear, penetrate it, and the innards are just going to disperse, and then your body's going to dispose of it. Things like penicillin, for example, which is what this example was. However, depolarization, same, but different. Create these channels within the outer membrane of these microbes. Then you start having a positive and negative imbalance, and you ruin homeostasis. We, as humans, our skin, our internals, need to maintain a homeostasis, all of our cells, in order to survive. When that gets disrupted, things die. And that's what they found was happening. So kind of like a different way to attack based on all of the proteomes that were analyzed. And what they did was, just like any sort of deep learning, neural networks, machine learning, leverage a bunch of databases that I believe I have. Yeah, for example, the NCBI has extinct organisms that are represented with a sequence data of GenBank. So you can train these deep learning models, various ones. Theirs is called Apex that they created, and they have ones for bird, bony fishes, mammals, whatnot. And there were specific ones that showed interesting properties, like in this one, notably lead compounds from the woolly mammoth, from the straight-tusked elephant, from the ancient sea cow, the giant sloth, and the extinct giant elk showed anti-infective activity in mice with skin abscess or thigh infections. Molecular de-extinction aided by deep learning may accelerate the discovery of therapeutic molecules. So all of this goes into this model that they created called Apex. And here, I don't want to go into the details because, full transparency, I really haven't gone into the math and how all of this works in this neural net. I know that there's a bunch of data sets, much like this one from the NCBI. They have their own internal ones from the researchers. Let's see, this article was actually published in the Nature of Biomedical Engineering. And I believe if you go down to the bottom, they'll talk about exactly what data sets were leveraged, selectivity scores, how they used everything, the actual math behind it, and what they came up with. And then they also used a program. So all of this data has to be trained into something called, and this is open source, called PyMol. And PyMol takes all the data, allows them to sequence it, get these generative outputs where they can then analyze antimicrobial properties. That is what drug discovery, like how drug discovery is actually accelerating in the age of artificial intelligence. Another one I personally have had my hands on that I find interesting, NVIDIA has their own within what's called NVIDIA AI Enterprise, BioNeMo. You can deploy it with an enterprise environment, train it on, they have some pre-trained models, train it on certain data sets that you want to use. And there's things like AlphaFold from Google, for example, where you can then do all of the simulated drug discovery, how antibiotics are going to work. Very, very fascinating, interesting things. I just haven't seen where we want to look backwards at all of these extinct animals, sequence the proteomes, discover within them what will work. And actually right here, if we zoom in, we can see just simple graphic, proteome of extinct organisms. Here's the sequencing, deep learning based antibiotic prediction, again, using PyMol, all the different data sets that they can leverage, and then make informed, very highly sophisticated predictions based on a lot of probability and statistics, and then test it, experiment and validate within mice. Incredibly fascinating. This is the new age of drug discovery. What I'm hoping it doesn't turn out to be is just another way to create things that eventually lead to stronger microbes out there where antibiotics aren't working. Perhaps we can be very selective and more intelligent of how we treat ourselves, maybe even not treat. And that's something that we talked about as an undergrad in microbiology and virology. Is it sometimes okay not to treat? Let our bodies create their own resistance to them, become immune, fight things off, and that way we're just not making that next generation of highly resistant microbes and viruses out there.